Welcome, welcome. Here's our tutorial on codominance Punnett squares. Codominance is that fun one where we have two dominant traits that are both visible in our offspring. My favorite example, as you should probably know by now, are those roan cows. We get a dominant red cow mating with a dominant white cow. They breed together, and their children are what's called roan cows. They're kind of splotchy, patchy, red and white. What makes this different from incomplete dominance is that in incomplete dominance, we get a third new phenotype of a blend or a mix of those two colors from our parents. But where codominance gets interesting is that both of those two different alleles are present separately as themselves. In the roan cow example, red and white, they don't mix and they're both present. So let's take a look at some of these examples. I'm working on paper. It's easier for me to explain. Maybe it's easier for you to understand if you can see me writing as I'm talking versus just me typing on a computer. You can't see the process as easily. So if you're working on your computer, no problem, great. If you want to work on paper with me, totally great too. All right, let's get started. So tulips. Tulips are a flower. This is a bad picture. I only have a laser printer that prints black and white, but we can get creative here. Let's make it, so tulips can be codominant in flower color. Yellow, I'm gonna make this kind of yellowy with my highlighter here. Yellow is dominant, and let's just pretend that this pink highlighter is red. All right, yours should be on color if you have your computer open. But red is also dominant. Heterozygous tulips, we will get what we call the Spartan phenotype of yellow and red. Both yellow and red are present separately. We're not mixing to get orange. No, we have both yellow and red there. So question number one, what are the genotypes of the various phenotypes? Great question. So we're given the phenotypes. We got yellow. Let's make this a blue color. Yellow is dominant with Y. And we got red is also dominant, but we get the letter R. So we don't have to come up with any letters, not a problem. We don't have to get creative here. So what are the genotypes for these various phenotypes? Well, when we're talking about codominance, at least in my class, for the most part, we're just gonna stick with homozygous dominant. There may be examples like with blood typing when we do that in class, where we get some recessive things happening, but just to keep things simple, when we're talking about codominance for these examples, we're only getting homozygous dominant for our pure colors. In the tulip example, yellow and red. So what does that look like? All right, so yellow has the Y. It is dominant, so we're going to get a homozygous YY there. Both of them are capital. This one looks a little bit smaller, but it is also big. And the red is also dominant with the letter R. Cool. But then we also have this Spartan color where we have red and yellow present together. Well, when we have a heterozygote in codominance, we're going to get one of each letter. One of each letter. So our Spartan color will be half blue, or I guess blue ink Y letter, half pink ink R letter for red. Or if we want to do it this way, let's just make it YR. If we want to make a new phenotype, have a separate color. Up to you. Okie dokie. So number two, create a cross of a homozygous red with a heterozygous individual. So they gave us the phenotypes. What are the genotypes? All right, so individual one. Individual one is homozygous red. Homozygous red we found was RR. Cool. Individual two. Individual two is heterozygous. That means it's going to have one of each letter. I'm going to use purple because blue and pink together make purple. There we go. So we get a heterozygous Spartan flower for individual two. Now we'll plug it in, make a Punnett square. We don't have males and females listed out for us. We just have individuals 1 and 2. So I'll just put 1 across the top, 2 down the side. We're going to put the alleles in here, one allele per column for our individual 1, and one allele per row for our individual 2. Cool, cool, cool. Tight, tight, tight. Now we make our potential offspring. These 4 boxes here will be our potential offspring. We're going to do some Punnett squaring. Okie dokie. So we're going to get an R from 1 and a Y from 2. So that would be our purple color here. Actually, let's just stick with Y and R. Sorry, technical difficulties here. Y from the individual 2. Why do we put Y first? Because that was the one that was given to us first up here. And then R from the individual 1. Y from individual 2. R from individual 1. And for this one, we're going to get a R from individual 2 and an R from individual 1. You could do either one here. I'm just doing this to keep my colors looking a little more consistent, and it ultimately doesn't matter. All right, now let's look at our genotypic and phenotypic ratios. How many of our offspring here are YY? Looks like none of them. So that'll be a big fat 0. How many of our individuals are RR? Looks like we got 2 of those there. And how many of our individuals are the heterozygous YR? We got 2 of them there. Whoops. Talking and writing 2 different things. So there is our genotypic ratio. You can write 2 out of 4, or if you like percentages, 50% and 50%. This one is 0%. All right, now we need to look at our phenotypic ratios. I am going to use some colors just to make things fun. I'm going to use these 3 here. All right. So the yellow flower will be yellow. Look at that, easy peasy. The red flower will be this pink color, and Spartan color will be, let's just say, this orange highlighter. All right. So how many of them are going to be yellow? Well, according to our genotype, none. How many are going to be red? Well, looking at our genotype here, our R stands for red. We're going to have 2 of them are going to be red. And how many are going to be our Spartan heterozygote here? We got 2 of those. Boom. 2 of those. For our ratios, you could say 2 out of 4, 2 out of 4, or if you like percentages. There we go. We did it. We did our first codominant cross. All right, our next one here, number 3, we're going to cross a homozygous red individual with a homozygous yellow individual. Okie dokie. So we're given our phenotypes. What are their genotypes? Homozygous red. Homozygous red. Well, we know from all the way up here, homozygous red is big R, big R. And our yellow individual here is blue in the past. Homozygous yellow is going to be our individual 2. So looking back up here, we know yellow is YY. All right. This is the problem with working on paper, is that I've run out of room. I have to do a little switcheroo here, keep things as organized as possible. All right. There we go. Cool beans. All right, so now we need to make our Punnett square. Individual 1, I'm just going to put across the top just because it is going there first. Then individual 2, 2 big Y's. Awesome, awesome. Now we're going to fill in our offspring. 1 from each parent. We're going to do Y's first because that was the information presented to us. Ultimately, it doesn't matter. It's just a preference from person to person. All right, so we're going to have a Y from individual 2, an R from individual 1, a Y from individual 2, and an R from individual 1. Over here, a Y from individual 2, an R from individual 1, Y from individual 2, and an R from individual 1. Cool, cool, cool. Now, we're going to look at our genotypic ratios. Slide this up a little bit. We need to figure out how many of each of these genotypes we see in our offspring. How many YYs do we see? Looks like a big fat 0. How many RRs do we see? None. Another goose egg. How many YRs do we see? Well, I count 4 of those. If you like percentages, this is 0%, this one is a 0%, and this one is a 100%. Cool, cool, cool. Now let's take a look at our phenotypes. Just like the last one, I'm going to mark the yellow ones with a yellow highlighter, the red ones with a pink highlighter, and our Spartan color here with an orange. Now, going back to the first page, the Spartans are our heterozygotes. Going back, it's like manual scrolling on a screen. All right, so looking at our Punnett score, we have all of these YRs, which we figured out here in our genotypic ratio. If we have YR, that means we're going to be Spartan. Orange, 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 orange highlighter. Perfect, perfect. So none of them are yellow. We know this because of our genotypic ratio. None of them are going to be red because we know because of our genotypic ratio. And looking at more genotypes or even our Punnett square here, we know that all of them will be Spartan colored. They get one allele from each parent. 100% are going to be the Spartan color. 0% for yellow and red. All right, that is all I have for codominance. I hope that was helpful.